We are going to welcome our second keynote speaker, Peter Lansdorp, sort of like, one of like the cool leaders of the telomere field, developer of amazing methodology, and my eyes are kind of bad, so I'm going to hold it out here. So Peter Lansdorp is an MD PhD, so here's to the cool MD PhDs, people who spent a lot of extra time in school. I appreciate that. Is a distinguished scientist at the Terry Fox Laboratory in Vancouver, and a professor in the Department of Medical Genetics and Hematology at the University of British Columbia. From 2011 to 2016, he was the first scientific director of the European Research Center for the Biology of Aging of the University of Groningen in the Netherlands. Did I say that right, Simone? Groningen? All right, I got it. See, and I can't read anymore, but we'll get there. He has made numerous, like a million contributions to the multiple research fields, and has published more than 281 papers that have been cited more than 50,000 times. Okay, that's a lot of being cited papers. The focus of his current research is further development and applications of the single-cell strand-seq method developed in his laboratory, along with other lab methods, to study genome assemblies, haplotype, resolve structural variations in human genomes and DNA repair. And so we are incredibly lucky to have him presenting today, and look forward to lots of information about really thinking critically about methodologic development and its relationship to our goals in the Telomere Network. So take it away, sir.
Thank you very much, Stacy, for that very kind and generous introduction. I'm sorry I'm not in New Orleans. I know you were planning, or you're having a great party there. On the other hand, I don't miss the COVID tests and the embarrassments of air travel and all that. And I must say I've been really enjoying this meeting. I'm particularly happy actually with the presentations from Rita Efros and Simon Verhulst yesterday, which touched upon a lot of points that I will be revisiting. So I think that was a great, and all the discussions that I've heard, I've learned a lot already. So I think it's been really good. So thanks again for having me here. All right, so yeah, I'll give you I couldn't resist on putting the first slide in here, if I can. Why doesn't this? Yeah, which is my summary of what I've heard so far. There seems to be a very strong desire to make accurate measurements, which are precise, and but I think the tools, particularly these amplification-based tools, PCR, are challenging to do that, right? So I think and there was a comment made by Simon yesterday. Well, I mean, maybe TRF, like it's done in Avi's lab, is great, but we cannot all do that. Well, in reality, that may be the challenge here, that we have measurements which are accurate, which cannot be done by everybody unless you really want to invest the time and equipment to pull that off. So I want to make this statement that Einstein made: everything should be made as simple as possible, but not simpler. And I'll be touching upon some of the critical issues there related to the technology that my lab helped develop. I was actually also very pleased to see that, well, before I say anything further, I should mention that I have a conflict of interest because I founded a company to make clinical measurements. So anything I say can and will be held against me, I'm sure, but I think the flow-phase data that I'll show you have some of the accuracy and the precision that I think it would be very useful, but unfortunately the method is not really easy or cheap, or so it's. But you can learn more about that company if you want to. So actually, Stacy tried to twist my arm to talk more about method development, but the truth is that I haven't done any telomere length measurement development for the last decade. So I'll be revisiting old data, and some of it may be new to the new people in the field. Some of it will be very familiar to actually. It was pleasant. I'm very happy to see so many names that I remember from my telomere past, and then even collaborators, and I'll mention some later. But I'll go over what I know about telomeres, But more and more, I'm now interested in actually evolution biology and telomerase, because I think that with the new data actually coming from sequence, basically measuring mutations in DNA, we have a better, at least I think I have a better idea as to what actually might be going on, and why we have these differences between the mice and between rodents and humans, for example, in terms of telomere biology. Okay, so before I forget, because it's always at the end, it might be tricky, I want to mention that what I'm going to talk about today really is the result of collaborations through the last 3 decades, I guess. A lot of work was done in my lab, but I couldn't have done it without a lot of people that are listed here, and I'm sure I'm forgetting all kind of people. But the bottom line is that this, what I'm telling you is not just my work or something like that. This is really an effort from a large number of labs in great collaborations. And again, I was very pleased to see actually Gabriella Berlocher, who played a key role in developing Flowfish on this meeting, as well as Sharon Savage, which is also, I think, a valued collaborator. And then there's lots of people here, which which I just want you to have a look at, which also contributed. Okay, so there's an interesting twist, which reminded me very much, and I put some extra slides in based on what Rita told us yesterday. I was really at the early 90s. My main interest after my medical school and the PhD actually was looking at hematopoietic cells. So I was intrigued by the possibility to use monoclonal antibodies to dissect this enormous cellular diversity that we know exists in bone marrow, where we have continuous production of cells, billions of cells produced every day, T cells, B cells, but primarily, of course, granulocyte and primarily red cells. And they all come from common precursors, which ultimately are derived from hematopoietic stem cells. And you can see in this slide here a number of arrows. And I want to make the point here that I think people typically underestimate the depth of the hierarchy. So there's a number of cell divisions. So the stem cells, I believe, divide less than once every 2 or 3 years, maybe even less than that. But they generate precursors, which divide a little bit more frequently, etc. So you have a very deep hierarchy, which limits the number of cell divisions over a lifetime to something that's very close to the Hayflick limit. So I think that goes against the current dogma about cell turnover, where people like Hans Clevers would claim stem cells in the mouse divide in the gut every day, or maybe in humans every 3 days. And people like Bert Vogelstein, esteemed colleagues, but they talk about billions of stem cell divisions, whereas I actually think that Hayflick was right, and I'll try to make the arguments as we go along. But anyway, I didn't know that at the time. In the early 90s, I was actually very keen on figuring out this self-renewal property. So my skills at the time were was that I could make monoclonal antibodies. So we made actually a lot of different monoclonal antibodies, CD34, CD90, CD45 or 8. They're still in widespread use, and they can be used to really dissect this primitive compartment of CD34 positive cells. And these were the days when the old kind of growth factors, cytokines, were cloned, became available. Serum-free tissue culture media were set up. So I was really keen to figure out how to push these stem cells to self-renewal, because I realized that what you need for stem cell transplantation, and we heard a nice talk this morning, are actually stem cells. And we still typically like to use donors, but if you could purify the stem cells from a patient and figure out how they would self-renew, you could do that. So that was really the goal, but that actually failed miserably, up until the point that we started to say, well, maybe it was meant to be that way. Typically, we were working with adult bone marrow cells, but then, stem cells are produced maybe early on, and we started looking at earlier during development. And sure enough, so here on the left here, or on the right, you can see here what we found in adult bone marrow. So we could purify what we called stem cell candidates, and they would produce a lot of cells, and we could sort out the CD34 cells again, and they would again produce cells. We could repeat that, but the bottom line is that we couldn't observe a net increase in the number of primitive cells, CD34 cells. So, but if we then go to cord blood or even fetal liver, now we could see several hundredfold increase in CD34 cells, or several thousandfold. So I got very, actually I was stuck at that point, but made the observations and the paper, we published a paper, Journal of Experimental Medicine, good good journal, that stem cell renewal is developmentally controlled, but where to go from there? Well, at that point, so this must have been 93, actually I got a visitor from a student by the name of Human Faziri, and this Rita actually mentioned him. And actually he showed the data that he did in collaboration with Rita. These smears, so he actually asked me, did you look at telomeres in this situation? And I looked at these smears and I thought, well, I'm not sure what this all means, but you know what? I'm going to send cells blindly to Human Faziri, who was actually at the time, I think, working with Cal Harley and Rick Elsop they had just were in the process of moving to Geron company. So I sent the blinded DNA samples from fetal liver, these cultures at different time points. So I couldn't produce enough cells for sudden analysis because that's what they were doing. But we could produce enough cells to basically look at what was going on in terms of telomere length. And I really got interested in telomeres when it was found that actually in these cultures, they started with bone marrow, the telomere length was clearly shorter, and with each subsequent passage, there was a decline. And the cord blood was kind of in between bone marrow and fetal liver. So I published this paper, which I think is not quoted a lot. I don't know why. I think it's because people don't think about stem cells in the context of telomere biology. But this to me made the point that there's evidence of a mitotic clock in hematopoietic stem cells, which of course goes against this notion of self-renewal, which is still propagated. And I'm sure at this meeting, there's people believing that you need telomerase to maintain stem cells so they can produce stem cells or blood cells over a lifetime. If they assume that there's more than 100 divisions of these cells altogether. So, these smears, they were supporting that there is a clock ticking, a mitotic clock in stem cells, but we could produce a few hundred of these cells. So I got really very interested in better ways to look at telomere length. And at the time, nice work from Moises et al. Had shown that you can actually use in situ hybridization to detect telomeres. In fact, they described that the telomeres in human cells, or I guess all vertebrates, were TTA, GGG. So, and I was kind of interested to look at the single molecule level at telomere length. But as you can see here, you can see these are metaphase chromosomes. So you would expect that the two sister chromatids show the same signal, but you can see there's a lot of variation. So, and actually when I asked around at the time, so this is 94 to, Dan Pinkel or Barb Trask maybe means something to some of you. I mean, these are experts in situ hybridization. They said, well, Peter, forget about in situ hybridization for quantitative purposes, because you need a few kb and then you can see a signal sometimes. And that was actually more or less what you see here. So but as luck would have it, I got my hands on a new type of molecules that were synthesized in the University of Aarhus in Denmark by two chemists, Peter Nielsen and Michael Ekholm. So these were people making synthetic molecules. I mean, and they came up with a structure that is a kind of a DNA mimemic, you could call it, where normally in DNA or RNA, you have a phosphate-based sugar backbone. And they described the molecule where you have an aminoethyl glycine backbone, more like a peptide-like backbone, where bases are attached in a way that they can base pair with complementary DNA. So they figured out the stereochemistry of synthetic probes that could base pair. And the big difference between PNA and DNA you can see here is that this peptide-like backbone has no charge. I mean, in DNA and RNA, you have a phosphate group which gives kind of a negative charge, in which you need to overcome for in situ hybridization. So you typically use high salt to allow this base pairing to happen in the presence of these repulsive negative charges from the backbone. But I figured out, well, maybe we can use, and that was, of course, I had discussions with Michael Ekholm about this. Maybe we can use conditions where DNA cannot renature or DNA cannot hybridize, but PNA can. And so I actually started during a sabbatical in Leiden, started using these probes and figured out that in fact you can make that happen by conditions that are now used by anybody, I think, using fish probes or telomere probes, which is basically means low salt. So you basically can do hybridization. So the principle would be that you would denature DNA, typically using heat and formamide, and then you would do that in the presence of an excess of probes, which are directly labeled with a fluorescent dye. And then you would basically allow the probe to hybridize, which would now, under conditions, again, low ionic strength, so no possibility of re-annealing, so that the probe can hybridize. And then you can analyze. Well, we focused initially on microscopy, and I'll say a few things about that, and then we'll talk about flow cytometry. So that's the principle, and that's I think been very powerful in all kinds of studies. So we for the first time now we could see telomeres on every chromosome end, and we can also measure the total content of repeats bound on cells if you hybridize in suspension. Maybe I should move straight to the next slide, because you can now, of course, do karyotyping. So we preferred for these fish studies to look at metaphase chromosomes. There we have a measure of the two sister chromatids, so we know we're dealing with differences, because you would expect these two sisters to have more or less the same amount of DNA. And you can see that there's large differences. So if we do karyotyping, chromosome 1 here, you can see there's lots of telomere repeats on this particular homolog, but very few on this one. And you can see other chromosomes like 17 here, which has very few repeats on the P arm. But in general, you can see large differences between chromosomes. In fact, that was the title of the first paper we published here on this method, is heterogeneity of telomere length on chromosome arms. It's not like, and in fact, we then spent a lot of time, actually, in the decade that followed, to perfect these techniques, realizing that the microscopes, fluorescent microscopes, are certainly the ones that we were using at the time, are primarily qualitative instruments. They're not designed for quantitative purposes. There's issues related to the illumination field. There's issues that relate to breaching. There's issues related to focal planes. And you have to get this all right if you want to have any hope to do any quantitative measurements. But we spend a lot of time with using plasmid calibration. So we took indeed plasmids with defined inserts. And I can attest to the fact that it's difficult to have large inserts in E. coli plasmid, or if you want to propagate plasmids in E. coli. But we could actually develop more or less quantitative measurements by taking images at multiple planes, using piezo focusing devices. A lot of technology went into this, and then of course we had the issue of how do you now analyze these images. And I was fortunate that I could rely on Stephen Poon, who came up with segmentation algorithms to basically find the telomeres, find the chromosomes, and then basically calculate the integrated fluorescence per spot. So we could actually start doing quantitative measurements on telomeres of metaphase chromosomes. And what you can see actually again is this heterogeneity. So the length here, and we measured here, we calibrated this for telomere fluorescent units, so that relates to the amount of repeats that we could measure using the same approach on plasmids that we deposited on slides. But yeah, the bottom line is that for every chromosome, there was an enormous range, but the averages do differ between chromosomes. That's maybe shown nicely here. So here we look at 10 different individuals. We look either at fibroblast or at bone marrow or at peripheral blood cells, but all metaphase chromosomes. So we cultured these cells typically to harvest the metaphase chromosomes, and then we did this in site hybridization. And what you can see here is that, for example, in individual 4 and 5, chromosome 11p is very short, but in individual 3, both in fibroblast and in bone marrow, telomeres are very long. So there's heritable variation in telomere length on specific chromosomes. And you can see this similar thing for 16, which is very short in this individual, but pretty long on this individual. So, and interestingly, and that's been confirmed by Stella in Cardiff, chromosome 17p in our hands is one of the shortest. In some cases, it's indeed the shortest, but in other cases, in all cases, it's one of the shortest telomeres. Whereas, for example, the telomere length on chromosome 4q is pretty long in all individuals. So it would be very interesting to figure out what are the differences. Presumably, there's more likelihood of telomerase acting on 4q. Is that why is that? Is there more heat? less heterochromatin, or what we don't really know. I think what dictates these chromosome specific telomere length will be something for the future to figure out. But what we also saw, and I think this is important for thinking about environmental exposure to telomere damage and maybe replicative lifespan and biological parameters, is that what we saw, if we just looked at clones of cells, and we in this case focused on chromosome 17 and chromosome, I think this was the X chromosome, I'm not quite sure, but what we did see is that in some cases, whereas most X chromosomes, the P arm here, you can see nice, easily detectable signals. In some cases, you suddenly bang, you see very short repeats. The same for 17P. Here, the P arm, or here the Q arm, you see a kind of a dramatic loss. There still was some these were fibroblasts, so there was still some detectable telomere repeats, which was probably needed for these cells to get to the metaphase states. But I think that's you have this stochastic variation, which I suspect relates to DNA damage, maybe to some real issues in terms of repair at the telomere, where if there's a stalled replication fork somewhere inside the chromosome, the stall may be rescued by a fork coming from the other end, whereas maybe that kind of backup mechanism or repair of stalled forks is not available to telomeres, which are inevitably replicated from internal origins. Okay, so I think that makes a point here that I think we have to consider this stochastic variation or stochastic loss of telomere repeats in our models of telomere loss and how it relates to biological properties. So it's possible that, and this is, I guess, from a slide in fibroblasts, it relates to this Hayflick limit. So early on, and this was data actually, well, it's not from the big figure I stole here, is not from the paper but Jones studied in the 80s. He basically took fibroblasts and split the pairs, and he looked for the replicative potential of the individual daughter cells, and he found that there's a large variation. So some daughters would stop very soon, and others would continue for a large number of times. But the bottom line is that as these cells march towards senescence with population doublings, there's fewer and fewer cells dividing. So the bottom line is that it takes longer and longer to get a population doubling, and in the end, there's in fact very few, if any, or in the end, the culture is called senescence, and there's no more dividing cells left. So, and what we found when we looked at in fibroblasts and different population doubling. So here we looked at population doubling 10. So these were early cells. I think these were embryonic fibroblasts, but I'm not sure. Again, we've observed between clones large differences in replicative potential. So some clones would go 20 doublings, others would go 50 doublings. But the bottom line is that as they march towards senescence, there's an increasing skewing of the telomere length distribution. So it could still be relatively normal at an early passage, but there seemed to be selection of at least a minimum number of telomere repeats as these cells march towards senescence. So telomere loss, and of course, in fibroblasts, they may express very little telomerase, maybe a little different in hematopoietic stem cells or lymphocytes, where there's some telomerase, so that the ability to rescue some of these short truncated telomeres might be higher in those hematopoietic cells compared to a fibroblast. But the bottom line is that we have to take these stochastic losses of telomere repeats into our models, into our models of mitotic clock or the Hayflick limit. So based on that, I would suggest that indeed I'm also very interested in new methods that measure the length of telomere repeats in single DNA molecules, because I do realize that with flowfish or by Southern, we typically come up with a median or an average length, and we're missing out on these important, and that was even discussed this morning, these important short telomeres which may dictate the fate of a cell. So we would really like to look at the distribution of telomere length next to the average length or the median length in cells, and ideally also DNA samples. But I think that raises other problems that have been extensively discussed here. But I think there's promise. I mean, you can maybe look at telomere length measurements of individual molecules using combing, And I'm also interested in, and I'm happy to hear. I want to learn more about that by listening to other talks. There might be possibilities for parallel single telomere Q digital real-time PCR. So to look at how signals develop over time to get a sense of the actual length. What I think is important is to of course include controls. Well, we'll talk about that later. So the next few minutes I want to talk about flow fish. So it's possible, and let's just go straight into it. So with a lot of effort from multiple people, so Nathalie Rufer, Tim Brumendorf, I mentioned Gabriella Barlocher. I mean, we perfected this technology. So just for those of you who don't know how this works, we get a blood sample. It needs to be relatively fresh, more than 2 days old. Cells will degrade, but just like for your DNA extraction, whole blood is a good source of DNA, in this case cells. So we then lyse the red cells, so we end up with nucleated or leukocytes. We add some bovine thymocytes, which we use as an internal control. I mean, that's an important point. If you want to do a quantitative measurement, you need to have some kind of calibration point. You wouldn't want to do a Southern without molecular weight markers. Well, same for these fish measures. You need to have some kind of internal control. And of course, by flow cytometry, where you measure thousands of cells, you can add these cells in. And by using cow thymocytes, which are very long telomeres, and they are very small, so we can distinguish basically the signals that we get if we just look at the whole hybridization probe procedure without a PNA probe, or the procedure with a PNA probe. And we can establish for each cell type, which we can gate. So we can gate on the thymocytes. We can see a very low background fluorescence and a very strong signal. And then with lymphocyte, we can see things, and we can see even gate specific lymphocytes, like in this case, CD20 positive cells. And you can see that actually some cells there, memory B cells, very long telomeres, but in lymphocytes, in fact, they're typically, well, you'll see what we found there in case you haven't looked at our papers yet. But there's differences, and these cells here, these are memory T cells, and the bulk actually are more naive T cells. So this is what you get if you, for example, look at a 5-year-old and a 9-year-old. But again, I'll show you there's a lot of variation between individuals at any given age. So again, we calibrate for this with these cow thymocytes, and you can see that there's a large drop in telomere fluorescence in the granulocytes, which have a high side scatter. And for the lymphocytes, you see actually that there's some cells which have still more or less the same telomere length as the granulocytes, but you see this large population in this 9-year-old individual, T cells which have much shorter telomere, and these are in fact mainly memory T cells. So that's actually shown here. Now we've done this on hundreds of individuals, enormous amount of effort. Again, Gabriella should be credited with other people in the lab to actually pull this off. But there's some very important points that can be made from this slide already. First of all, it's not so clear here, but the largest drop in telomere length is in the first, I would say, 1 or 2 years of life, when there's a lot of cell divisions at the stem cell level, but also at the lymphocyte level. But the decline continues quite rapidly till the end of puberty. But by then, telomere length in lymphocytes is much shorter than what you see in granulocytes. And in granulocytes, which we like to use as a surrogate marker for the turnover in stem cells, assuming that there's a fixed number of cell divisions between a stem cell and a granulocyte, and realizing that there's billions of cells produced from a single stem cell. And again, there's a very deep hierarchy in my view about that explained that. But you can see that telomeres, particularly in lymphocytes, are getting very short. As a courtesy to our friends that use Southern analysis, actually, we plotted all these results relative to what you could measure by telomere restriction fragments, or TRF, by basically adding 3 kb to the actual measurements. But I'm now more or less convinced that we should just forget about that and just should measure. So you should think about that this is actually the absolute amount of telomere repeats if you just subtract 3 kb from these the values I show here. So what we also found, which is interesting, is that telomere length differ between the sexes. So females have on average longer telomeres throughout life. So that is already seen at in umbilical cord blood. So if you look at different, you look at lymphocytes or granulocytes, and again, these are not large numbers, but there's reproducible differences. It is very significant if you look at, for example, naive T cells here over a lifetime. And in fact, here you see these dynamics of different cell types, which I think were mentioned by Rita yesterday as well. So lymphocytes go down much more quickly, especially memory T cells, which a lot of immunity building up in the first during the first few years of life, and we rely on these memory cells, but we might be running out of them. In fact, we might be running out of NK cells as well. But if you look at this difference between males and females, in fact, you could, that is of course very simplistic, and I realized that you could argue, well, how accurate are these regression lines? But data is published. You can repeat this analysis. You would say, well, that's good for maybe 5 to maybe up to 10 years of life expectancy differences if telomere length were really dictating differences in life expectancy altogether, which of course is debatable. Well, we have heard some interesting discussion about longitudinal data. We were actually very interested to look at longitudinal data from especially in after birth, because we saw this big drop. Is that real? So we would be hard pressed to convince parents to have their blood sample from their baby every 2 weeks, but we could convince ethical committee that this would be valuable information. So we did this in collaboration with the Primate Center in Texas, Karen Rice. So we actually obtained blood samples every other week from two newborn baboons. You can see there's actually very good longitudinal data. You can see this drop. It's much more pronounced in T cells than in granulocytes and B cells, which seem to run parallel. But there's a remarkable drop in this turnover, which we think is turnover primarily of stem cells at the age of 1 year in baboons. And we think it's probably very similar, maybe 2 years, maybe 3 years in humans. We haven't really done enough of these measurements at these early age. But of course, you need accurate measurements to do this. I actually doubt you could get the same kind of results by Southern, but we'll talk with Avi about that later, I guess. So I also want to make the point that now this baboon colony may have been selection, there may be weird things going on, and we just looked at a handful of animals, or a few dozen maybe, but there's enormous differences between the animals, right? So here you have, it's definitely still baboon, but its telomere length in granulocytes and B cells is 25 kb, whereas in other animals it's half. So the start length, and of course the decline then in the first year, is much more pronounced in this animal than in this. So I think this absolute telomere length is fraught with challenges and difficulties, because there may be just, the length may be set early during the development, and there may be large differences between animals, and maybe more in animals than in humans actually, but they may be important in how things play out during life. And of course, I realized that we can use these telomere length measurements in patients which are deficient in or have telomere biology disorders. In this case, we're haploinsufficient for telomerase reverse transcriptase or the RNA gene compared to the siblings. And what's really striking is that these individuals have telomere length which are way below the first percentile of the normal distribution, which you can see here. So this is actually then useful, because quite often a clinician is faced with the challenge of how to treat these patients, and compatible siblings are a good candidate for a bone marrow transplant. But we've reported cases where they use the sibling with the same haploinsufficiency, and then that marrow graft would fail, and that would of course cause terrible suffering in the families. And anyway, so it's useful for clinicians to actually measure first of all, measure telomere length to see whether there's a problem at the level of telomeres, and secondly, decide whether they want to use a sibling for a transplant, if that is even a consideration. So, and I would point out that this, of course, we're talking about haploinsufficiency of telomerase giving rise to fatal disorders in humans, whereas in the mouse, in fact, when I got involved by I was soliciting collaboration with people like Carol Greider, and I also, Rhonda Pino. I never heard of them. Actually, Maria Blasco, I eventually, say, yeah, we would be interested. They have submitted this paper. I think Cell wasn't all that keen because it didn't make sense. They could just knock out telomerase. We all had thought at that time that telomerase was very important, but it took generations. But we could show actually by Q-phase that you lose 5 kb of telomere repeats with each generation. At very late generations, you see a beautiful phenotype of telomere fusions and infertility and all kind of problems that actually resemble somewhat, but not completely, the phenotype that we see in people that are haploinsufficient or telomerase deficient. So why is telomere biology in humans and mouse so different? So I guess I got interested into that more and more. And I frequently are reminded of this quote from Dobzhansky, with nothing in biology makes sense except in the light of evolution. So I started to look into what we know about biology of senescence, and that was kind of interesting because there's a lot of interesting stuff published in that area. And in fact, the first, I think the most important contributions were made by this person, August Weismann. So if you think about evolutionary biologists, the first of course, Charles Darwin comes to mind. He's probably number one. I would say a close second is this person, August Weismann. This is way before DNA was even recognized as a molecule of heritability. He basically, by careful observation, proposed that there's a distinction between the soma and the germline, and he proposed that the germline is potentially immortal, whereas the soma is mortal. And he made the point that the heritable material is transmitted only through the germline, and the soma has nothing to do with that. It's called the Weismann barrier. Well, he got a lot of flack for that because people would like to believe, especially in communist countries, that the environment would dictate what's happening to the offspring as well. But he also, in the context of telomere biology and I guess evolutionary biology of senescence, he made some very important and I think prescient observations. And he proposed that in terms of aging and senescence, we're dealing with mechanical wear as well as a cell division limit. He said, these are two things that contribute to senescence. Well, then the second, I would say, great evolutionary biologist was George Williams, who in 1957, and actually Rita mentioned this here, that he invented this idea of antagonistic pleiotropy. So whereas genes that can enhance fitness early in life often have detrimental effect late in life. And in fact, they're expected, there is no selection against it, so you might expect them to accumulate. But George, although he wrote that paper in 1957, he didn't mention DNA, although that was published a few years ago, but we all suffer from being kind of myopic and focusing on our own little territory. But he actually criticized Weismann, and he basically made the point that this Weismann, he's a clever guy, but he's got it completely wrong because it's fallacy to identify senescence with mechanical wear. And of course, we've looked for decades for mechanisms that would support some kind of cell counting or death mechanism. We haven't been able to find it. And of course, he was wrong in both counts because we now know that DNA damage is the primary cause of aging. So we accumulate mutations every day as we, in every cell. And loss of telomeric DNA is being recognized now as a bona fide mechanism that limits the number of times cells can divide. So the third evolutionary biologist that I want to mention here is Thomas Kirkwood. He, in 1977, basically took, I think, Weismann's ideas about the germline and the soma, that these are separate lines. And he made the point that senescence results from mechanical wear in the disposable soma theory of aging. And the essence of that theory is that the amount of energy that an organism allocates for somatic maintenance and repair is less than would be required for the soma to last indefinitely. So, so the germline has better protection against DNA damage and against mutations than the soma. And this theory predicts that damage would accumulate, somatic damage would accumulate throughout life and contributes to aging and ultimately death. And I would think, and this is, I think now it's got really interesting and important, because this is all history, but this, or old history, we now get into recent history, because there's very strong support for this theory from recent sequence data, which shows that cells of the soma accumulate mutations at a rate that is tenfold higher than what you see in the germline. So let me just, I want to particularly highlight this paper here. This is work from Marty Corena and Peter Campbell in the group in the Sanger Institute in Cambridge, England. They've basically perfected the method to look for mutations in DNA. And they basically looked at mutations in different cell types, and they compare the number of mutations that you can see in stem cells, which you can do by sorting CD34 cells, growing them up, do whole genome sequencing and count the number of mutations, look at the signature of mutations, but do the same thing with granulocytes, which are divided by at least 20 cell divisions or more. And basically what they find is that the correlation is there's basically perfect correlation between the mutations that you see in the stem cells versus the granulocytes, and they share the same signature. And these mutations accumulate almost linearly in time. So there's mutations per cell. You can see that again, here's this stem cell versus the DNA. You actually, this is a summary here. You can make the first point I should make is that sperm cells, so the cells of the germline are better protected against this accumulation of mutations than any somatic cell. But interestingly, cells of neurons, whether they're dividing or non-dividing, they accumulate mutations at a much higher rate, which is typically tenfold higher than what you see in the germline. So that goes against, and it certainly went against my notions about how mutations originate in DNA. We always felt that cell division was the most important thing, but I think based on this work, we have to conclude that it's DNA damage which results from metabolism and maybe exogenous damage as well. I mentioned this in the context of telomeres. So we have thousands, tens of thousands of lesions, DNA damage events per cell per day. 99.99 something are repaired very efficiently, but everything, as clockwise, things slip through. And of course, that's so if you're 80 and you've got accumulated 40 mutations per year, you've got 3,000 mutations per cell. Individual mutations are different between one cell and another. That's likely to compromise function, and that's likely to contribute importantly to aging. But in the context of living longer, this allows some arguments about putting a limit to cell division by limiting telomerase activity. So the point here is that indeed I think we have to agree that there's disposable soma. We all subject to disposable soma. So mutations accumulate a tenfold higher rate in the cells of the soma compared to the germline. This mutation rate is similar between dividing and non-dividing cells, surprisingly. So your heart cells, brain cells, gut cells, blood cells, they accumulate mutations at a similar rate. It's a very surprising finding, actually. So errors during DNA replication, cell division, are not the major cause of most somatic mutations. And these mutations are time dependent rather than cell division dependent. So if you want to live longer, you better get your cell divisions in early, and you better figure out, put a limit to the number of cells time cell divide. I should mention, and there's more stuff that you can now read about the mutations, because there's some tissue specificity. Mutations that you get in the brain are slightly different than in other tissues, presumably related to specific metabolism or DNA repair pathways available to those cells that needs to be resolved. But if we now think about cancer, and mutagenesis is primarily time dependent, inevitably mutations will occur. And again, I want to highlight that this is time dependent. But if a mutation occurs in a cell that can divide, like a stem cell, Of course, the daughter cells will also have this mutation. And if you now imagine that these cells have to want to live longer, the chance that they will acquire a second mutation increases, etc. So there's a link between the need to suppress tumor cells if you want to live longer, and I think the ways nature has found a way to resolve that is by putting a limit, or by basically mutating the promoters of telomerase and suppressing telomerase expression in somatic cells. So concentration of stem cell division early in life, suppression of telomerase levels in somatic cells links to the lifespan of animals. And that's actually been supported in rodents who typically don't, well, there's large variation in lifespan. But also Woody Wright and Jerry Shea, who was on this call, so we can maybe talk about it later as well. They made the point that there is a correlation between telomerase suppression and lifespan. This is just a mechanism. It's apparently easier to put a limit to the lifespan than increase the efficiency of DNA repair. We can talk about that, but I think that's the drugs, the key of one of the key new messages that I'm hoping to propagate here. So restricting the reproductive lifespan or reproductive potential of cells may have provided a selective advantage during evolution that allowed lifespan to increase by suppressing tumor development. But as Rita mentioned, and as I mentioned with Williams, he had this notion that mutations or alleles that are beneficial early in life often have detrimental effect late in life. And I think that's also true for telomere shortening. So I put this all together in a theory that I'm hoping to publish soon, is that is which I call telomerase erosion in disposable soma. So the TETS theory of aging. So the clockwise accumulation of mutation in somatic cells is likely to compromise the function of an increasing number of cells and contribute to aging. This, I mean, people always argue, well, why do mice get older and there's no telomere shortening? So what do telomeres have to do with aging? Well, I think that, you know, in human aging we have two factors: this telomere shortening and this accumulation of mutation. Whereas in the mouse, it's presumably primarily this accumulation of mutation that's causing aging. The disposable soma theory proposed by Kirkwood does not include a limit to the number of times cells can divide, as was proposed by Weisman. So I'm actually proposing to include that for human aging. And limiting telomerase levels in somatic cells allowed lifespan to increase by acting as a tumor suppressor mechanism. So while advantageous early in life by suppressing tumor development, telomere erosion contributes by aging by compromising cell renewal late in life. And I think we have to particularly think about cells of the immune system, but also cells of the cardiovascular system. And of course, we know that immune deficiency and cardiovascular problems are major causes of mortality and morbidity late in life. So I think that that needs to be taken into account. So that's the main message here for my take-home message, is that I would like to discuss and propose that DNA damage as well as telomere erosion cause aging in long-lived animals like ourselves. So just to make this point a little bit more graphic, I guess I've shown you data to show that there's a distribution of telomere length, that at birth T or lymphocytes are longer than what you see in granulocytes, but at the end of puberty that's already reversed and it's more pronounced even at the 60-year-old. I now actually show the actual length. So and I would make the point that it's a good thing to have telomere shortening because it prevents cancer early in life. It's now a lot of data supporting that having longer telomeres is more often seen in cancer-prone families. But the flip side is that, you know, you probably have more immune failure or cardiovascular problems late in life as a result of that. And cancer is somewhat of a different story because by allowing more cell divisions, having more reproductive potential, the chance that you accumulate mutations and that these mutations will be propagated in cells that can still divide increases. But if you have very short telomeres, there's also loss of stem cells, and that puts high pressure on renewal of remaining stem cells and provides strong selection for abnormal cells. So cancer, unfortunately, you know, you're really in between a rock and a hard place if it comes to telomere length and cancer late in life. And interestingly, we did publish, and I just, you know, I didn't try to polish this slide, but we did publish in 2008 or 9, I can't see actually what, is that if we looked at super senior, so these were collaborator Angela Brooks Wilson here in Vancouver, looked at people 85 years and older, never had diseases, cancer, very healthy individuals. And we looked at telomere length, and indeed the telomere length on average was shorter, but what was striking in that the variation that we see at any given age seemed to be much less. So maybe that supports, but these numbers are too small. I realize that there's some selection of an optimal length, a Goldilocks, if you want, in terms of telomere length. And of course, then you could argue, well, why, if telomere shortening evolved to suppress tumors, we're dealing with a dismal failure because we do get, of course, cancer is a major cause of mortality as well. And I think it has to do with the failure of this mechanism where, the limit is not imposed properly. And why is that? Because on the one hand, there's very strong selection for cells that ignore DNA damage response from short telomere. So I would argue that loss of p53, which we see in most tumors, is a reflection of that failure of that tumor suppressor mechanism. And then the other is that, with intact DNA damage responses, you might get fusion of short telomeres, which will then fool a cell into dividing with fused chromosomes. And I show sisters, but it could be other chromosomes. And you might actually then end up with cycles of fusions, breaks, and bridge cycles, which can cause aneuploidy, which in fact was already proposed by people very early on in the telomere field. Okay, so I think I've provoked enough ideas here and response from you. So I'll just want to summarize. I'm suggesting that progressive loss of telomeres in somatic cells with age is implicated in aging and suppression of tumor growth. The telomere erosion in disposable soma theory of aging predicts that clockwise accumulation of mutations as well as telomere shortening contribute to aging in long-lived animals. Telomere-mediated loss of stem cells provides strong selection for cells with defective DNA damage responses. And new methods, and I guess this is to Stacy, new methods that measure the length of distribution of individual telomeres next to the average length promise to improve correlation between telomere length and biological variables. So I'll stop there and take some questions. Thank you very much.
Thanks, Peter, for a wonderful talk and very stimulating ending, in particular because this is not the way I expected the talk to develop at all, but it's wonderful. I have, of course, there are lots of questions, and I won't abuse having the microphone in my hand for too long. The first one is that you mentioned when you said, okay, you want to include Weismann's idea that there's a limit to the number of cell divisions in the disposable soma theory. You wrote that in the note, but then you added verbally to explain evolution of aging in humans, and but that wasn't in the text. So I'm curious how you see that.
Yeah, not so much humans, Simon. I would include a long-lived animal, so maybe even birds and dinosaurs. I bet you they had suppression of telomerase as well, and they had telomere shortening to suppress tumor growth. So I think there's just limitations in terms of DNA repair in the soma. I mean, animals that would spend more energy and had better DNA repair would probably would be eaten by the animals that don't do that. So that's, if I said humans, I meant long-lived animals.
Yeah. Okay. And secondly, on this thing that has just been made very small, the second point you put there is that you say you make a prediction, but aren't these actually the assumptions on which you
particularly prediction do you?
Well, the second point that you predict that clockwise accumulation of mutations as well as telomere shortening contribute to aging along animals, but you base your theory on the observations that this is happening. So aren't these sort of assumptions rather than predictions? So I'm curious what new predictions.
I guess the phrasing can definitely be debated. I would like to exclude that. There might be other things going on as well, right? I mean, I'm not saying that because I didn't discuss, I guess, structural variations that or transposon insertions, other things that might be contributing to aging as well. So, but I think the two main things are this disposable soma theory, which is not generally accepted, I guess, and the telomere erosion, and the combination of those two indeed is that, yeah. But I think you have a trouble here with the language, which can probably be phrased a little bit more accurately.
But leading on from that, so did you think about possible new predictions on the basis of this theory and the observations on which it's based?
Yeah, one prediction is that is for example that we now know that the people over 70, have 20% of people over 70 have clonal hematopoiesis. So they have a clone which has some kind of mutation which gave it a proliferative advantage. And my prediction would be that there will be clones that will disappear as well, because now the mechanism actually works. And I haven't seen any papers. Maybe there's some hematologists on this call. Maybe they can. I know it has been described in PNH, but I suspect that some of the clonal hematopoiesis that we see, and again, this is very prevalent in, again, 20%. Well, there's people would argue that everybody over the age of 20 will have some kind of clone with a mutation, but the size may be small enough, and there may also be. Anyway, my prediction would be that we will see some more disappearing clones in normal humans.
Okay, thanks. I'll go see who wants the microphone next.
Hi, Waylon Hastings here. I have a question. So earlier this year, Alexander Wolf had his paper on the tumor suppression theory of aging, where he pointed out that only those mutations which lead to increased proliferation are important for driving aging. And I wanted to hear your thoughts on that as it relates to this telomere erosion and disposable soma theory.
I need to read that paper. I know that in terms of I can think of gain of function mutation as well as loss of function mutation that may provide stem cells with a proliferative advantage, right? You may lose tumor if you alter the cell cycle, or if there's so I need to really look at the paper. It's one of the things I'm only recently started digging into literature, and I came all the way till 1977, so I haven't looked at at all the papers in this field. So I'm better off not commenting at this point.
Hi, thank you for that talk. This is Kalen Ryan. I think first of all, I agree with you as, Darwin, Weisman, and Williams probably being some of the biggest thinkers in evolutionary biology. But it seems like one of the things I've struggled with Williams is antagonistic pleiotropy theory for aging is the idea that changes that happen early in life, for example, a function of a gene can't change throughout life. So we know that a lot of genes do one thing early in life and something else later, or their expression levels go from high to low or vice versa. So I think it makes it challenging to figure out why something like telomerase couldn't be active early in life and less active later on, or vice versa, or whatever the case may be. So I'm wondering, in the context of your theory, why protection against cancer earlier on what the constraint might be between having the best of both worlds, I guess, is what I'm asking, between early life and late life.
Yes, and I would agree with you that, of course, genes can have multiple functions, and they might be different between early in life and late in life. I think your point is probably well illustrated by what's happening in B cells, where in the germinal center, there's naive B cells coming in with a increasingly shorter telomere length with age, but they come out with longer telomeres, and that's presumably because it's important evolutionarily to allow for affinity maturation and selection of antibodies with a particular sequence. So you have a lot of cell death, but also bottlenecks in terms of clonal proliferation. But the flip side there is that by loosening restraint on this telomere shortening and allowing cells for more cell divisions, you might have more B cell malignancies than, for example, T cell malignancies in humans. So I think, yeah, biology will go wherever or evolution will go wherever it can go. And if it makes more sense to lose a restraint or to use a different function of a particular mechanism of gene, it will do that, as long as it can get away with it. So I think it's a blind watchmaker indeed.
Thank you.
No. Okay, now I'm on. Dan Eisenberg here. I'm really struck by the variation in telomere length across the different chromosome arms. And one of the things that I've found most striking and very rarely commented on is that the size of the chromosome arm seems to strongly correlate with the length of the telomere. And I think this is evidence in humans and other species. And I'm wondering if you have thoughts on the biological and evolutionary significance of why we see that such a strong correlation between chromosome length and telomere length.
Do we see that? Let me just stop. No, sorry.
I think other studies have shown it more. I don't know if it was evident if you were directly analyzed in your slides.
I don't recall this, to be honest. So this is the slide, right? So this is what we published. It's all I know, really. And this is the P arm, and this is the Q arm. And you're making the point, that on average here the P arm is shorter than the Q arm, or what is the?
So in other studies where I've seen people analyze the telomere length in relationship to the millibases of chromosome arm length, and I've seen really strong correlations, I think like 0.5 or 0.8, somewhere around there, between the size of the chromosome arm and the length of the telomere. And apparently I thought this was reliable findings, but maybe you're not familiar with it.
Well, we've seen that in the mouse, but in the mouse the P arm is very, of course, mouse chromosomes are acrocentric, and they're very close to the telomere on the P arm is very close to the centromere, which is presumably more heterochromatic. But I would say this is the most reliable data you're looking at. I don't know any other data in humans, and if it argues against what we are seeing here, I would very carefully look at that data, because I think there's a lot of noise in the whole telomere field. I mean, it was nice to see actually that that this finding has been confirmed by Stella, so that also in the hands of Duncan Baird, 17p happens to have fewer repeats on average. But again, there's a lot of variation, and I have to agree with you that I'm puzzled by this enormous variation, right? So is there even more variation? Well, these were, I guess, yeah. So it's striking how much variation there is. There must be a minimum that's needed for. These were all, of course, metaphase chromosomes, all cells that were allowed to divide. So I don't know, but yeah, maybe again, I just definitely don't claim that I've seen all the literature. I have been busy with other things in terms of actually the single cell strand sequence is really exciting lately. So maybe if there's good data, but I'm inspired by Simon's talk and talking about accuracy, and I think there's a lot of noise in the literature, unfortunately, related to telomere biology and telomere length.
This is Professor Sika. Very interesting talk. Really enjoyed it. You earlier mentioned that telomere length is larger than in the female than the male. Now the question intriguing is to my mind, is there any correlation to that extent that the woman mostly has less prone to heart disease, cancer, and have a longer lifespan than the men? And if it is an early indication, I would suggest maybe these palmistries people and horoscope readers should measure telomere length than anything else.
Yeah, it's a good question. I would refer to Aviv, who is, or Dr. Abraham Aviv, who has done a lot of work on this here. I guess women on average, they live longer, but they eventually also, a major cause of death is cardiovascular disease. So how that correlates exactly, I don't know. I mean, what I think is very interesting is why do we have these differences even at birth, right? So, and I have a hypothesis about it, but I'm not going to talk about it today.
So I'm intrigued by this idea of rapid early proliferation being sort of protective against later DNA damage. And I want to couch it, and I'm going to rope Alyssa in that one of the models that we think about in terms of the relationship between exposure to early life adversity and early life stress is this early acceleration of aging. And that exposures to adversity, trauma, violence early in life are much more impactful in terms of these age-related health conditions such as cardiovascular disease than those same exposures later in life. And I am curious about that because we also have this idea that there's that selective advantage early that comes at a cost later. And so I'm wondering what your thoughts about that in terms of the model of the relationship between both shorter telomere lengths and early life adversity and stress, but also this sort of protectiveness against sort of DNA damage later.
Yeah, a really good
Peter
question.
first.
Yeah, I don't have answers because we haven't really looked with this flow phase method, for example, at effects of stress. I do believe that the oxidative damage or other damage to telomeric DNA is a reasonable hypothesis to put forward related to accelerated telomere shortening, but there might be effects specifically on lymphocytes versus, for example, granulocytes. So it would be really interesting to look at that in the context of specific cell types, which I think has been missing in a lot of the data or the studies that I've seen or read superficially and a long time ago, I should add. So
Thank you.
I cannot say more about that, Stacy. Sorry.
No, that was a good answer. This is Alyssa now, and I'll just say that the other pathway that we're not thinking of or talking about is the impact on stem cells and the early shaping and pruning of the immune system is quite dramatic. And so I think these early traumas, we know there's thresholds and under 5 matters more, et cetera. So if we were able to measure telomerase and telomere length in stem cells, that might be part of the mechanism. So, a little bit less about circulating cells.
Yeah,
and rather setting the tone for how much, proliferative potential throughout life. And then the other pathways, which any psychologist here is probably thinking about, which is that it's changing the emotional and behavioral pathways, the persistent risk pathways that last and have effects throughout the lifespan toward higher stress reactivity and poor health behaviors.
I hear you, but I don't think you need, I don't want you to start looking for stem cells because they're very rare, even much more rare than all the papers that you've seen, because there's probably only 100,000 cells in the bone marrow, which can be called the stem cell, and you have this very deep hierarchy. And you can use granulocytes as a surrogate marker what's happening on stem cells. I think it would be very interesting to compare telomere length in lymphocytes and in granulocytes in people who've been exposed to different types of stress. I think I haven't seen any data on that. I'm not sure if there's some TRF data. I'm suspicious of a lot of the PCR data.
Well, we have PCR data, Peter. Putterman et al. Looked at both granulocytes and lymphocytes, and there was a stress effect on both types of cells, which surprised us because certainly we, were more excited to see more sensitive effects in the fresher granulocytes, but or less of an effect since they were more short term, but we saw no difference.
Okay, well, interesting.
I have a comment on Zoom from Michelle Heacock. She said, I may have missed this in your talk. Does the observation that 17p is among the shortest telomeres and 4q among the longest correlate in females and males?
again, we've looked only at 10 individuals. There's a lot of variation. These differences between males and females on average length are so huge that you would need to do a much bigger study. But I suspect that on average, females will have shorter 70 or will have longer 17p than males, and that these differences will hold. But I have no data to support it.
So thank you for a wonderful morning session and excellent discussion, and raised a lot of really focused goals for the afternoon. So we are going to take a break here and eat really good Louisiana food. I hope wherever you are, you're going to eat somewhat not as good food, but equally good, nutritious. And Peter, we're going to get you down here as soon as we can. So thank you again, and thank you everyone for a great discussion. We will be back at 1.
Thank you, and I'll be happy to join the afternoon. Thank you again.